Proteins are going to be perhaps the most detailed form of biological molecule, and it's not really nice to put it this way, but the way I see it, proteins are going to be perhaps the most important type of biomolecule. It's a controversial statement, so I'm not going to further elaborate on that, but the way I see it, without the amino acid structure, without the amino acid infrastructure, basically all living beings are going to fall apart, the way I see it. So I would say proteins are basically the most or close to the most important biomolecule. Therefore, we have to learn about this in a tad bit detail. Starting off with its composition, when it comes to proteins, we have the usual carbon, the usual hydrogen, the usual oxygen, and we have nitrogen. Plus, sometimes we can have phosphorus or sulfur in them, but they're not considered, phosphorus or sulfur, they're not considered as being main constituent elements, whereas the CHON, carbon, hydrogen, oxygen, nitrogen atoms, elements are considered as being constituents of proteins. The protein monomer is the amino acid, and these are very versatile. AA for short, sometimes A dot for short, like so. Let's look at an amino acid. Let's learn how to draw an amino acid so that you can draw an amino acid even if you are blindfolded. There are different ways to draw an amino acid. You might have practiced a different way. If you know how to confidently draw an amino acid, stick to that. That's fine. But if you have not yet acquired that skill, you will after this. You have to follow things through as I explain in the exact same manner. That way it's going to be easy. I think I'm going to pick a darker shade of purple. Ooh, nice. Okay. So it's like this. You start off with a central carbon atom, C. Okay, start off with a central carbon atom, C. And then on top of that, you have a hydrogen atom. Nothing hard, nothing complex. You just have a hydrogen atom on top. That's okay. On the right hand side, you have a carboxyl group. What exactly is a carboxyl group? A carboxyl group is the C double bond O OH thingy. That is a carboxyl group. On the left hand side, you have something known as an amino or amine group, which is simply NH2. And finally, this is the variable chain. We typically call it R, the variable side chain. That's going to help you go a lot. Let's actually label these things. First, this is typically, sometimes this is also referred to as the alpha carbon, okay? The main carbon, simply nothing special, the main carbon. Let's label these things. That's basically, by the way, that's everything of an amino acid, if you wondered. So this is a hydrogen atom, okay. This is what you refer to as a carboxyl group, or sometimes a carboxylic acid group, but call it a carboxyl group. This is an amino or amine group. Amino or amine group. And this is the variable side chain. That's what you call it. Due to this, only can different amino acids exist. So we have 20 different forms of amino acids, either way you pronounce it. The only way that those 20 different versions are possible is because the side chain is different. All these things in all those amino acids are the same. They have an amino group, an H atom, and a carboxyl group along with an alpha carbon in the middle. This thing is what changes. We have a variable side chain. Now that we know how to actually draw an amino acid, it's important to understand how they behave. these amino acids are joined via a certain specific type of bond known as a peptide bond. Joined via peptide bonds. Peptide bonds. Like so. How exactly is a peptide bond born? Let's see. Recap on how to draw an amino acid. Central carbon, alpha carbon, you have a hydrogen atom on top. Carboxyl group like this. NH2 amine or amino group, and then just the R side chain. Also noteworthy mention, this can sometimes be drawn like this. The OH, come on. OH being here, the oxygen being here, and the bonds basically angled like this. So I'm going to keep it like that then for a bit, like so. And for a peptide bond to form, just like a glycosidic bond, you at least need 2 amino acids in total. Let's form the bond. So simply OH combines with H. What are we getting out of this? That's going to be a water molecule. I'm going to get rid of all of that. And those are the bonds that are remaining, and the bonds are just going to connect. The way you draw the final peptide is like this. I'll just draw that part only. C double bond O, and then you take the N and you can place it like this. It's only one NH, no, this thing. It's only one NH, and then it's back to this carbon. That there is your overall peptide bond. That is your peptide bond. That is your overall peptide bond. Simply speaking, that would be your peptide bond. Sometimes this can also be drawn like this, where the H just comes on top and the bond is here. That is also perfectly fine, but this has a little bit of a flair to it, does it not? Right. Whilst discussing about all the biomolecules, now let's talk about the structural arrangements of the most important, I mean, according to me, the most important biomolecule. Let's talk about protein structure. Let's talk about protein structure. Proteins have 4 specific structures. The first structure is a 1-degree structure, simply primary structure. The primary structure of proteins simply refer to the sequence of amino acids in a polypeptide chain. In a polypeptide chain. Then we have the secondary structure. So if you were to visualize this, it's simply the primary structure of proteins refer to the sequence of amino acids in a polypeptide chain. Okay, for you to form a protein, you're going to need amino acids that are bonded by peptide bonds in a long chain. The primary structure of any proteins, there are 4 levels, there are 4 degrees, there are 4 structures, I mean 4 levels of structures of proteins. The first primary structure is the sequence of amino acids in a polypeptide chain, like this. Then what exactly is the secondary structure? Secondary structure. The secondary structure is the hydrogen bonds of the polypeptide backbone, hydrogen bonds of the polypeptide backbone, causing cause the polypeptide chain to fold. To fold in certain shapes. Here's the deal. We have this amino acid sequence now. This amino acid sequence can fold in all these different varieties. Fold in all these different varieties. The main two ways in which the secondary structure hydrogen bonds of the polypeptide backbone can fold is two. The first one is referred to as an alpha helix, and this is similar to amylose. See, that is what you call an alpha helix. Then you can have something known as a beta pleated sheet. A beta pleated sheet. That looks something like this. Beta pleated sheets look something like this. Beta pleated sheets. So primary structure, sequence of amino acids. Secondary structure, this sequence can fold on itself because of hydrogen bonding. What are the two main structures they're going to form? That's going to be an alpha helix and a beta pleated sheet. And I just noticed there's the E missing here, polypeptide backbone. Primary done, secondary done. Tertiary structure is where things get interesting. Tertiary. This is where three-dimensional folding begins. Three-dimensional folding of the polypeptide chain. Due to the side chain reactions. You remember we talked about the side chains, right? So these side chains can undergo certain reactions with each other, with other molecules, and some other forms of things. That's how tertiary structures are formed. It's due to the formation of excessive bonds by the side chains, which are the R chains, thus giving the polypeptide a distinct structure. Now, what exactly are these side chain reactions? These side chains can undergo certain types of bonding. Due to certain types of bonding, these side chains can change. We have four main types of bonding. A possible question would be, what are the four different types of bonding under the tertiary structure of proteins? That's a possible question. So number one, I think I'll use pretty pink, or should I use this? I'll use this. The first thing is we can have hydrophilic or hydrophobic interactions. Let me write that down. Number one, hydrophilic interactions. Hydrophilic or hydrophobic interactions. How does that work? Hydrophilic or hydrophobic amino acids, they can orient themselves as they wish. AA short for amino acid. These amino acids orient themselves towards the center of the polypeptide or away from it to avoid or not avoid what? Right, how does that work? Let's talk about hydrophilic first. Hydro means water, philic means to like. Okay, hydrophilic Hydrophobic means scared of water, so does not like water. So hydrophilic means likes water. Hydrophobic does not like water. Does not like. So simply speaking, by using general knowledge, you'll see that any hydrophilic amino acid will orient itself towards water because it likes water. Any hydrophobic amino acid will orient itself away from water because it does not like water. The second kind of bonding that can occur is an interesting type known as a disulfide bridge. So disulfide bridges can occur. Now this is about a very specific amino acid. This is about an amino acid known as cysteine. The amino acid cysteine contains sulfur in its structure. That hence the name disulfide bridges, because there can be bonding or bridges that can occur between these sulfur atoms. The amino acid cysteine forms a bond with another cysteine, so that's the disulfide bridge, another cysteine through the R group, because cysteine is actually, sulfur is actually present in the R group. So you have a disulfide bridge forming from cysteine to cysteine. We typically represent it like this. So something like this, a disulfide bridge. And now previously we had hydrogen bonds, no? In the tertiary structure, sorry, in the secondary structure. Then again in the tertiary structure, we can have hydrogen bonds again as well. So here, polar R groups form bonds with other polar R groups. You know, when it comes to hydrogen bonds, it's going to be between two polar molecules, no? For example, if we have water like this, water is going to be delta positive and delta negative, like so. So there's going to be this interaction between positive and negative. Is a very vague representation, but that's basically the idea of a hydrogen bond. Polar groups are going to form bonds with other polar groups. That's what's going to happen. The last kind of bonds that you're going to be able to observe would be ionic bonds. Ionic bonds. Ionic bonds can only occur on charged R groups. Can only occur on charged R groups bound to each other. We have to have a charged side chain for all this to work. Otherwise, it's not possible. And those are the main 4 types of bonds that can be involved in the formation of the tertiary structure of proteins. With that, we can move on to the final quaternary structure, the 4-degree structure, the quaternary structure. It's very simple, very straightforward. What color did I use for this? It's green, right? Eyedropper green. Okay. A quaternary protein is simply a protein consisting of more than one polypeptide. That's it for quaternary structures. Right. Now I gave you the 4 main levels of protein structure: primary, secondary, tertiary, followed by quaternary structure. This was just information. There were visuals as well, but I'm going to visualize things right now. Starting off, before everything, we have this. What is that? That is our trusty old amino acid. This guy can form peptide bonds like so, and now what we have is an amino acid chain. That is a primary structure, primary level of protein structure, the chain of amino acids. Then when it comes to the secondary structure, these amino acids, due to hydrogen bonding, can take different forms. Can take different forms. I'm doing my best to draw an alpha helix and failing miserably. You get the point, right? That is like a brain twisting moment. So this is what I'm trying to draw. Alpha helix, or they can form the beta pleated sheet. Like so. It's either the alpha helix or the beta pleated sheets at the secondary level. At the tertiary level, due to complex bond formations, these amino acids can bend, chain can bend in complex ways and form a very complex tertiary protein like an enzyme. Do you see? This thing's growing, this polypeptide chain is growing into a specialized 3D structure. Growing into a specialized 3D structure. Specialized 3D structure. Right. That looks like an enzyme, right? Okay. Let's pretend it looks like an enzyme. That is your tertiary structure. The reason why I wanted to visualize this like this was because you see, this is the same polypeptide. I've used orange and orange only to get up to this point. Primary, secondary, tertiary, we only have orange. This is where most students go wrong when they talk about the quaternary structure of protein. If we go back to the quaternary structure of proteins, you can see here, for a protein to be defined as being a quaternary protein, it has to have more than one polypeptide chain. More than one polypeptide chain. So this is just one polypeptide chain. Do you see? That's why even though it's a complex enzyme, it's just one polypeptide chain. If you want to make this a quaternary protein, this is what you have to do. I'm going to take it, copy it, place it here like this. Now I'm going to have another amino acid chain interact and wrap around this protein. Another amino acid chain interact and wrap around this protein. Give me a second. And yeah, there you go. That is a very beautiful, a pretty quaternary structure protein. Up until the tertiary structure of proteins, the same polypeptide chain, same kind. Once polypeptide chains start interacting, it becomes a quaternary protein. Now for this, you have another form of polypeptide interact. And this is very common in biology. interactions of polypeptide chains among. That's how basically we are built. This is how very extremely complex protein structures are born, like so. Different amino acid chains can interact and thus giving us a quaternary structure protein. Onwards to discuss about globular and fibrous proteins. Let's talk about globular and fibrous proteins. Globular and fibrous proteins. I'm going to draw a very simple table. I have no idea why this shift key is always giving up on me. And now that's gone, my lord. Give me a second. Okay. Let's draw a simple table. Whenever a comparison is born, try to go for a table. That's always good. So here I'll tell you the structures I'm going to talk about. Here let's talk about globular proteins. Here let's talk about fibrous proteins. First of all, let's talk about the overall structure. Globular proteins, when it comes to the overall structure of globular proteins, they are globular. Just like the enzyme we discussed here. They are globular. So they curl up so that the hydrophobic R groups point inwards and the hydrophilic ones outwards. Like we discussed before, it's the exact same thing. Hydrophobic ones will hide. Hydrophilic ones, they love water, no? They'll point outsides. Outwards from water, will be specific. From water. What about fibrous proteins? How exactly would they behave? How exactly would you expect fibrous proteins to behave? They are going to have long projected strands. They are not globular, they are not circular, they are strandish, forming long projected strands. The next property we'll talk about is solubility. And then we'll see what they do, what their specific roles are. Every time, I swear. Okay, roles are, and let's talk about a little bit of examples. What about the solubility of globular proteins? They are pretty soluble. But your fibrous proteins are insoluble. On your nails and your hair, you typically have fibrous proteins. Keratin is the protein that is fibrous. Does your hair dissolve? I hope not. Do your nails dissolve? I don't think so. What about the roles? Mainly, globular proteins are going to be enzymes. Mainly. Because of their hydrophilic and hydrophobic interactions, they're mainly going to be enzymes. And they're circular, like a ball curled up. No? So mostly they are going to have metabolic roles. For example, enzymes. Fibrous proteins like this, they are going to have structural roles. So hair, nails, collagen on your skin, that's to hold things up, no? That's because they have structural roles. I don't know why I use different colors for the two things. I should have used pink and blue, never mind. Some examples of each would be 1. Hemoglobin, something known as myoglobin. All of these things are to do with oxygen and transport in blood, and of course, enzymes. Like I said, for fibrous proteins, we have keratin and collagen, constituents of the skin and hair. Thus, we have a small comparison for globular versus fibrous proteins. Now, your syllabus requires that you know one example in a little bit more detail from each kind. So you have to know one globular protein and one fibrous protein in detail. The ones that have been picked are extremely good. You'll be learning about hemoglobin from the globular end, and you will be learning about collagen from the fibrous end. So let's start with the globular. I think I used pink for globular, right? Yes. Start off with the globular hemoglobin. It's soluble, of course. It has to be soluble, otherwise it's not going to work. It has a quaternary structure. When I say it has a quaternary structure, you have to straight away know, we have multiple polypeptide chains, multiple types of polypeptide chains, or multiple polypeptide chains of the same type. Either way, it can be quaternary structure. Actually, it has 4 polypeptide chains. Let's talk about the 4 polypeptide chains in more detail. 1, 2, 3, 4. No. First, what we have is something we call the alpha globin. And then we have a second alpha globin. Then we have a beta globin. Then, once again, we have another beta globin. Alpha globin, alpha globin, beta globin, beta globin. Alpha globin, alpha globin, beta globin, beta globin. Like so. 4 polypeptide chain. Now, here's the deal. To each of these I keep saying here's the deal too many times. I've noticed that myself. That happens on autopilot. I should probably stop doing that. I will. Each of these polypeptide chains is connected to a prosthetic heme group. Now, what do you mean by a prosthetic? Forget about the heme group. Okay, first of all, a heme group is simply iron, heme, okay? That's why it's called a heme group. What does prosthetic mean? Okay, if you don't have a hand or a leg of sorts, you're going to get a prosthetic. It's something that doesn't belong to you. That's not part of your body. Just like that for hemoglobin, prosthetic heme group is in the sense that iron is attached to the globin, attached to the polypeptide chain. It's not part of it. It's externally attached. That's the idea. For each of these globins, for each of the polypeptide chains, what's each of the polypeptide chain? Why do you always have to make this so hard? Copy and paste. There you go. Each of the polypeptide chains were going to have a prosthetic heme group attached. Boom. Like so. Alpha globin plus iron, alpha globin plus iron, the third beta globin plus iron, the fourth beta globin plus iron. That's the idea. Why do we have iron though? Can we not just stick to the globins and hope for the best? Yes and no. This is why we have iron. Now each iron atom binds to one oxygen. Each iron atom binds to one oxygen molecule. To one oxygen molecule. Therefore, a hemoglobin molecule can transport up to 8 oxygen atoms. I'll help you visualize that. Hold up. It's like this. An ion can bind to oxygen like so. So we have 4 ions, therefore 4 oxygens. That would mean that we have a total of 8 oxygen atoms in total. 4 times 2 is 8. At a given point, we are transporting 8 oxygen atoms. And if ion is bound to oxygen, your hemoglobin appears red. If ion is bound to oxygen, your hemoglobin appears red. If ion is not bound to hemoglobin, your blood or your hemoglobin, your red blood cells are going to appear palish. It's not going to be directly blue, but it's not going to be red. It's the idea. Okay, that's the globular hemoglobin part done. Now let's see what's going on with fibrous collagen. Fibrous collagen. Like we discussed, fibrous proteins are insoluble. Collagen is simply found in your skin, in your tendons, in your bones, and in your teeth. So it's mainly structure. Say main structural protein. A collagen molecule consists of 3 polypeptide chains. Consists of 3 polypeptide chains. So typically we refer to collagen as a triple helix. Say triple helix. Here's what it looks like. That's the first polypeptide chain. That's the second polypeptide chain, and that would be the third interacting polypeptide chain. And here, something interesting is in collagen, if you take account for every third amino acid, so I'm talking about every third amino acid, it's glycine. It's glycine. Every third amino acid is glycine. Right? That is basically fibrous and globular proteins complete. And thus, the protein segment is over as we speak. I think I will end it with this statement that I still think proteins are very important, perhaps the most important.